Hello and welcome back to Fluid Electrolyte and Acid-Base Balance. So the rest of this chapter we're going to be talking about acid and base balance, and I'm not really sure how strong your chemistry background is, so we're just going to start with a little acid-base review.
Acids and bases are everywhere. They're used to make foods, soaps and detergents, fertilizers, explosives, dyes, plastics, pesticides, even paper. Our stomachs are very acidic, our blood is slightly basic. Our proteins are made up of amino acids, and the letters in our genetic code, those A's, T's, C's, and G's, are all bases. You were probably taught how acids and bases behave on the molecular level. You were probably never taught that a long time ago, like ancient Greek ago, before anyone knew about atoms or molecules, acids and bases were defined by how they behaved. Acids tasted sour and corroded metals. Bases felt slippery and could somehow counteract acids. When molecules dissolved in water interact, they're exchanging two main currencies with their surroundings: protons, also known as hydrogen ions, and electrons. Depending on how a molecule is composed or shaped, it may be willing to donate or accept either protons or electrons with some other community member. And some molecules are far more aggressive than others when it comes to donating or accepting either currency. Remember that protons are positively charged and electrons are negatively charged. So if a molecule is willing to give up a proton, that's not too different from it being willing to accept an electron. Either way, it's becoming more negatively charged. Other molecules are willing to accept a proton or give up an electron. These are becoming more positively charged. Some substances are so aggressive about donating their protons that when they get a chance, all of the molecules in a sample will dump a proton, sometimes more than one, to the surrounding water molecules. We call these strong acids. Meanwhile, some compounds are so ready to accept a proton that they won't wait around. They'll just rip one off water, which usually has two protons but is generous enough to hang out with just one. We call these strong bases. Other acids and bases are not so strong. They may donate just a few of their protons to water, or accept just a few protons from water, but most of their molecules stay exactly the same. If left alone in water, they'll reach some equilibrium point, where maybe only 1 out of 100 or 1 out of 10,000 of their molecules has exchanged currency with water. As you might guess, we label these acids and bases weak, but in the common sense of the word, they're not weak. The vinegar in your salad dressing that you can smell from across the room, that is a weak acid. The ammonia you spray on glass for a streak-free shine, that is a weak base. So it doesn't take much to be an active player in the chemical economy. Most acid-base chemistry takes place in water, which can act as either an acid or a base, accepting deposits and enabling withdrawals like a 24-hour molecular ATM. And when a proton deposit customer, that's an acid, and a proton withdrawal customer, the base, shop at the same time, their net effects on water's account may cancel out, and we call this neutralization. Now, certain molecules can behave as acids or bases without water, but that's another story. Let's end by saluting water as the resilient and fair banker for acids and bases. It's always open for business, doesn't charge interest, and will never foreclose on your molecules. Which is more than I can say for wah wah.
Okay, so these are just some definitions. So an acid is a proton or hydrogen ion donor, and bases, in its simplicity, are a proton or hydrogen ion acceptor. Usually when you see an OH group or a negative charge, that's something that can accept a hydrogen ion. And like I said, usually you're going to see that positively charged hydrogen ion. So it's easier to recognize an acid. So just like that video was saying, there's strong acids and then there's weak acids, there's strong bases and weak bases. So something strong, that means it always dissociates into either the hydrogen ion acceptor or the hydrogen ion donor. For biology, we're usually dealing with weak acids and weak bases, except when we talk about hydrochloric acid in the stomach. But for today, we're going to be talking about weak acid-base pairs. So number one, we've talked about carbonic acid. It is a weak acid, and so there's always those arrows that kind of show that it goes both ways. I mean, sometimes it's going to dissociate, and sometimes it isn't. and then it can also reverse. So it's kind of at equilibrium between associated and disassociated. Just an example of a base, ammonia and ammonium. And just remember, electrolytes are ions in solution, so both acids and bases can serve as electrolytes. They conduct electricity. They'll complete that circuit. So the pH scale, that's really kind of the big thing I need you to know. And so the pH scale goes from 0 to 14, where the low end of the scale, you're increasing in acidity. When you go to the high end of the scale, you're increasing in alkalinity or basicity. Water is neutral because when it dissociates, is both the hydrogen ion, so it donates, but then this hydroxyl group can accept the hydrogen ion. So it's equal. And so the number just comes from negative the base log, so -7. So concentration, that's where it gets its number from. The big thing I want you to know is that we are going to look at acid-base balance, and I'm going to say that the set point for the body is 7.35 to 7.45. When we get outside of that range, that can lead to death. 7 is not the same as 7.35. And so there's a logarithmic change between one pH and the next. So between 7 and 8, for example, or 4 and 3, for example. So acid-base balance, the reason why we have to talk about it is because the pH is going to affect enzyme functioning, ion distribution, and the action of hormones. And so pretty much our physiology is made to function in this normal range of 7.35 to 7.45, mid being 7.4. A sustained pH below 7.35 is called acidosis. A sustained pH above 7.45 is called alkalosis. So how you can remember it, alkalosis has the K, so kicking up the pH. So 7.45 clinically, but we're going above 7.4, that's our set point. Acidosis has a big D in it, so the pH is down. Once again, if you've had chemistry and you have a good grip on this, then you are doing just fine. If not, here is your mnemonic. So acid, low pH, basic, high pH, or alkaline. So I told you that you were an acid maker. I told this to this unit. But where does all our acid come from? So now I'm going to prove it to you. We already talked about aerobic respiration of glucose. We get the respiratory acid or volatile acid called carbonic acid. When our skeletal muscles go through anaerobic respiration of glucose, they make lactic acid. We're going to get into metabolism, but when we break down our fatty acids, so that's through beta oxidation, we make acidic ketone bodies, so we make acids that way. When we break down proteins, there's some sulfur-containing amino acids, we make sulfuric acid. We eat other cells, so when we break down their nucleic acids or phosphoproteins, we get phosphoric acid. So all of our metabolism, we get acid, which is going to increase the concentration of hydrogen ions, which is going to lower the pH. So normal metabolic reactions generally produce more acid than base. And so we can have disease when those normal control mechanisms for controlling acid-base balance aren't working. Yep, that's all of us. So this is a great one for you to watch over and over again. So we are going to get into this regulation and this control of pH.
pH is an indicator of acidity. The body's blood pH is strictly regulated within a narrow range between 7.35 and 7.45. This is because even a minor change in acidity may have devastating effects on protein stability and biochemical processes. Normal cellular metabolism constantly produces and excretes carbon dioxide into the blood. Carbon dioxide combines with water to make carbonic acid, which dissociates into hydrogen ions and bicarbonate. This is an equilibrium, meaning all of the components of the left and right sides coexist at all times, and the concentration of any component is determined by that of others at any given moment. The rule of thumb is an increase in concentration of any component on one side will shift the equation to the other side, leading to increased concentrations of all components on that side, and vice versa. This equilibrium is central to understand acid-base regulation. Continued carbon dioxide production by all cells of the body drives the equilibrium to the right to generate more hydrogen ions. Because pH is basically a function of hydrogen ion concentration, more hydrogen means higher acidity and lower pH. Normal metabolism therefore constantly makes the blood more acidic. The body must react to keep the blood pH within the normal limits. This is achieved by two mechanisms. Elimination of carbon dioxide through exhalation. The amount of carbon dioxide exhaled by the lungs is regulated in response to changes in acidity. A decrease in pH is sensed by central or arterial chemoreceptors and leads to deeper, faster breathing. More carbon dioxide is exhaled, less hydrogen is made, blood acidity decreases, and blood pH returns to normal. Pulmonary regulation is fast, usually effective within minutes to hours. Excretion of hydrogen ions and reabsorption of bicarbonate through the kidneys. The kidneys control blood pH by adjusting the amount of excreted acids and reabsorbed bicarbonate. Renal regulation is slower. It usually takes days to respond to pH disturbances. Although all of the plasma bicarbonate is filtered in the glomerulus during the first step of urine formation, virtually all of it is reabsorbed back into the blood. Most of this reabsorption happens in the proximal tubule. The amount of reabsorbed bicarbonate in the proximal tubule is regulated via a number of mechanisms in response to changes in blood pH. It increases during acid loads and decreases during alkali loads. While the proximal tubule basically returns filtered bicarbonate back to the blood, the downstream collecting duct generates new bicarbonate by actively secreting acids. As protons are depleted from the distal tubular cells, the equation shifts to the right, producing more bicarbonate, which then exits into the blood. Hydrogen ions secreted into the lumen combine with urinary buffers, mainly filtered phosphate and ammonia, to be excreted in urine. The ammonia buffering system is particularly important because unlike phosphate, which is filtered in fixed amounts from the plasma and can be depleted during high acid loads, ammonia production is regulated in response to changes in acidity, and its concentration may increase several-fold when necessary. Blood pH is the main regulator of acid excretion, but potassium, chloride concentrations, and several hormones also play important roles. Pathologic changes may cause acid-base disturbances. Acidosis refers to a process that causes increased acidity, while alkalosis refers to one that causes increased alkalinity. It's not uncommon for a patient to have several processes going on at once, some of them in opposite directions. The resulting plasma pH may be normal, too acidic, called acidemia, or too basic, called alkalemia. Acidosis may result from inadequate function of the lungs, which causes arterial carbon dioxide to accumulate. This is respiratory acidosis. On the other hand, metabolic acidosis may result from excessive production of metabolic acids, ingestion of acids, decreased ability of the kidneys to excrete acids, or loss of alkali. Metabolic acidosis is characterized by primary decrease in plasma bicarbonate. Alkalosis can also be either respiratory or metabolic. Respiratory alkalosis is caused by increased ventilation, resulting in excessive exhalation of carbon dioxide. Metabolic alkalosis can result from excess loss of acids through the kidneys or gastrointestinal tract, bicarbonate retention, or ingestion of alkali. Metabolic alkalosis is characterized by primary increase in plasma bicarbonate.
Okay, so there was a whole lot on that video, and that's basically the rest of this chapter. So it's important for us to regulate our pH. We already learned that we're acid makers. So this maintenance of this balance usually is involved in eliminating the acids or at least buffering the acids. And so we have three ways that we can do this. We have chemical buffer systems, which they are going to be the fastest because they're already in place. We have the respiratory excretion. We know Whenever we exhale, we get rid of carbon dioxide and therefore that carbonic acid. Renal excretion, we know that we can get rid of hydrogen ions through urine, and that's why urine is usually acidic, pH 6, and we can conserve those bicarbonate ions. This is the slowest, but it's also the most powerful because we can get rid of all those other acids, so lactic acid, sulfuric acid, phosphoric acid, whereas respiratory, to be honest, we're just getting rid of our volatile acids, so our respiratory acids, so carbonic acid. Still good, but I'm just saying renal excretion is going to be the most powerful. So a buffer is a chemical or pairs of chemicals, which basically they just minimize changes in pH. They're not getting rid of hydrogen ions for us. They're just reducing the change in pH. So think about them. They're acting as chemical sponges that can either absorb hydrogen ions or donate a hydrogen ion. So usually all of these buffers are going to be a weak acid, weak base pair. So the bicarbonate buffer system, this is the one I said you needed to know this formula. We've talked about it already, but it's the most important buffer in extracellular fluid, particularly in the plasma. And so we've got these arrows that go back and forth, they're in equilibrium. And so here we have our hydrogen ions. This bicarbonate ion can accept that hydrogen ion, become carbonic acid. Carbonic acid, too, can also donate a hydrogen ion. So that's where we get our weak acid, weak base pair. So blood levels of carbonic acid are being controlled through the respiratory system. Levels of bicarbonate ion are controlled through the kidneys. So through that reabsorption of bicarbonate and that elimination of hydrogen ions. The phosphate buffer system, so hydrogen ion, monohydrogenated phosphate, so HPO4, dihydrogenated phosphate, so H2PO4. So this monohydrogen phosphate ion can accept a hydrogen ion resisting a drop in pH, and then this dihydrogen phosphate ion can then donate a hydrogen ion. So that's just where you get that weak acid-base pair. So this is a buffer. It is important in urine, which we just saw on that video, and in the intracellular fluid. The number one buffer and most plentiful buffer inside of cells, so in that intracellular fluid compartment, are going to be proteins. So proteins are made up of amino acids, so amino acid chains. So there's always going to be a carboxylic acid end and an amino end. And so I said acid, so a carboxylic acid can donate a hydrogen ion, but this amino end can accept it. And we've already talked about hemoglobin, so that globin part, that protein part, that is going to accept those hydrogen ions. When we get that chloride shift, we want to form that bicarbonate by that carbonic anhydrase. We kick out that bicarbonate and then we get that chloride shift. But we're still left with this hydrogen ion, and so that's what's going to combine with hemoglobin. And so that was just an example of a protein buffer system. So, and then two, the blood proteins, so albumins, those other globular proteins, they too can buffer acid in the plasma. And so protein buffer system, most powerful, most plentiful inside of cells. So big must know. So this is a big, here's a money slide for you. And once again, if you have a good background in acid-base chemistry, you're good. But if not, as long as you've got this, I don't care how you get there. Okay, so low pH means acid, means high concentrations of hydrogen ions, means bicarbonate is probably low, and CO2 is high, because we're going to tie this in the next episode. If pH is high, that means it's basic or alkaline. Those words mean the same thing. That means there's low concentration of hydrogen ions, usually brought on by a high bicarbonate. We get an increase in pH. So when bicarbonate is high, pH is high. Or there's low CO2, so carbon dioxide. Our set point is 7.4. There's an acceptable range. So below 7.35, it's acidosis. Above 7.45 is alkalosis. and then our one formula, so glucose or whatever food we've eaten, in the presence of oxygen, we go through that cellular respiration. Our waste products are carbon dioxide and water. Those two things will form carbonic acid. So carbonic anhydrase is the enzyme that will catalyze this reaction and make it go faster, but this reaction will happen on its own. And then carbonic acid is a weak acid, and so we get that bicarbonate ion and that hydrogen ion. So I'm hoping that you know all those things. So respiratory regulation of blood pH. And I've given you this mnemonic, so respiratory is opposite. And so what I'm saying is when carbon dioxide is low, that means pH is high. If carbon dioxide is high, that means pH is low. Because remember, carbon dioxide plus water, we get carbonic acid. So we know that by controlling rate and depth of breathing, we can adjust pH. So look right here. So acidosis, low pH, stimulates those central chemoreceptors, so brain and to some extent those arterial receptors in the aortic bodies and the carotid bodies. We're going to get increased rate and depth of breathing. So that's going to lower blood CO2. That's going to lower carbonic acid, and that is going to raise the pH, and so restore acid-base homeostasis. If our pH is too high, we call that alkalosis. That too is going to be stimulated by the brain receptors, can tell. Then we're going to get from the respiratory control center, remember the medulla and the pons, we get decreased rate and depth of breathing. So now someone's under breathing. So now someone is holding on to their CO2, and we said whenever CO2 goes up, that means pH goes down. So that's because if we hold on to CO2, we get carbonic acid. That's going to lower pH, and then acid-base balance is restored. So if you can kind of keep respiratory as opposite, you can keep track of your low CO2, that means high pH. High CO2 means low pH. So let's look at renal control of pH. So we know it is the nephron's job to filter blood and make urine, and that while they're doing this, they can regulate our hydrogen ion concentration of body fluids. Most of the time we are making acid, and so at the kidneys can excrete hydrogen ions into the urine. They're going to be buffered by ammonia, and then they can also reabsorb those bicarbonates. So just high intake of proteins is one way that we can generate acid. So when we metabolize those amino acids, we get sulfuric acid and phosphoric acid, and then increased concentrations of hydrogen ions in body fluids, so pH goes down. Increased secretion of hydrogen ions into fluid of the renal tubules, so into the urine, so then we urinate that out. So this should help restore our concentration, so lower that concentration of hydrogen ions in body fluids, and then we get homeostasis. So I've given you this mnemonic and I've given you this picture. So Rome, so respiratory is opposite, we kind of covered that. Metabolic is equal. What I'm saying is wherever the bicarbonate is, if bicarbonate is high, that means pH is going to be high, or pH is going to go up. If bicarbonate is low, that means pH is low. So come over here. So we've got acid-base homeostasis disturbed. So pH is low, so that's acidosis. So how the kidneys can respond to that, they're going to reabsorb bicarbonate back into the blood. So we're going to raise bicarbonate. So come over here. If we raise bicarbonate, that increases pH. And that's good because we were responding to a drop in pH. We also know that acid, that means the hydrogen ion concentration is high. So the kidneys can also excrete that in the urine. And so either way, we have acid-base homeostasis restored. If pH is too high, that's called alkalosis. So we can get rid of bicarbonate. And so if bicarbonate goes down, that means pH goes down. So if we're responding to high pH, we can lower the pH by lowering bicarbonate. The kidneys can also reabsorb hydrogen ions. And so the kidneys, that's why they are the most powerful in restoring blood pH. So they have this double whammy. So they can do it through bicarbonate and through hydrogen ions. So hopefully you got everything you needed there. We just have one more episode. Hang in there. Come back As always, if you have questions, I'm here for you. You have a great day.